Today, I'm going to discuss a fascinating and yet extremely complex field of this gut-brain interactions. And this is a quite provocative title, and the underline and the juice and the conclusion is that we don't know too much about this. Actually, we don't know a heck of it. So that's the bottom line. And matter of fact, you know, this is also the consequence of something that in this 2 days that I spent here been a recurrent theme, how now multidisciplinary approach to complex diseases like Autism Spectrum Disorder is a must. And again, if you look at, you know, the paradigm of, you know, the gene-environment interaction in multiple complex diseases like, you know, autism, you realize that, you know, the players are many. Definitely there is a genetic component, there is this intestinal component, there is the body fields where, you know, a tremendous amount of work is done to distinguish between friends and foes. There are a wealth of environmental factors that really play the game. But, you know, now we start to appreciate that this parallel civilization, complexity we call the microbiome, that has a great deal to do in terms of this yin and yang between tolerance and immune response. And this eventually will impinge with the clinical outcome that really can affect any tissue or organ, including the brain. And the question is how you connect the dots, how you really understand this complex network that really brings to the clinical fruition. And again, in order to really appreciate this, the key elements, particularly when you talk about the gut as the crossroad where all these elements occurs, is to really have a clear understanding definition of the gut and the gastrointestinal system in general. I have to say, even being an expert in the field, because I'm a gastroenterologist, there is no general agreement. In the past, very simplistic, we believed that the gut was a magic tube that transformed food in poop, and that's it. And then we start to appreciate how complex is the system, does much more than digestive, absorptive, you know, function, and so on and so forth. So if I have to give you a definition, really, of what is the complexity, the function of the gastrointestinal tract, and how this impinge in, you know, this balance between tolerance and immune response, and why has to do, or why crosstalk with the brain, I have tremendous difficulties even to find the right definition. The best that I can provide to you come from one of my students. I believe that she really nailed this down. She's a second grader. Forget about the spelling. So the intestine is a long tube with a clean opening on the top and a dirty opening on the bottom. This is the quintessential of what the gut is. I didn't get this fish business here until I inquire and I ask her, what is this fish all about? This is the noise that Dad makes from the dirty hands, she told me, and I met the fellow. So all this to say that, again, this is an organ that's been vilified many times because it's smelly, it has, you know, unpleasant content, and so on and so forth. But again, we come to the appreciation that is very, very important. The father of physicians already appreciated that the gut had too much to do, so much to do to so many conditions, to the point that Hippocrates many, many moons ago said all disease begins in the gut. I personally have revisited this, and this is my definition. The gut definitely is not like Las Vegas. What happened in the gut does not stay in the gut. That's a quote that I came in 2010. Now, this really has to do with this concept of gut-brain axis and how there's this crosstalk all the time. But, you know, the reality of the story is that, again, if you start to appreciate the complexity of the GI tract and you understand intestinal mucosa is really a battlefield. They're every day, 100 times a day, friends and foes needs to be distinguished. And the immune system of the gut, that is, by the way, the most complex immune organ that we have in our body, is called upon this decision-making so many times. And one mistake in that will have dramatic consequences. If you treat a friend as a foe, we will not sit here and discuss, you know, about science, because we will not survive without nutrients, for example. By the same token, if you treat an enemy as a friend, that can have tremendous consequences. So how the gut can really attend this complex and extremely important role in understanding and balance between tolerance and immune response. Tolerance meaning this is a friend, can stay. Immune response, this is a danger signal. I need to get rid of this because this can be dangerous. Now, sometimes the answer comes from looking into nature and how nature builds the business. And you don't see a squat there, and that's all right, but I will guide you. This is a cross-section of a gut. Happened to be of a mouse, but this is exactly in the human beings, the chimpanzees, you name it. And this is the lumen. And the first thing that you see is this single layer of cells, these epithelial cells, that is the intestinal barrier. They really protect us from the indiscriminated passage of these enemies that are passing through this GI tract all day long. You see immediately underneath the layer of cells, there are these green cells, they are the dendritic cells. First line of defense, if you consider, the immune system as sort of army, these are the soldiers that are sitting on the wall of the city looking outside and make sure that if they are enemy, they prompt the entire business to be ready to fight. Sometimes they send periscopes out, pedestals that they sample whatever is in the lumen, and if they perceive, danger, together with some receptor on the epithelial cells, they call pattern recognition receptors, in other words, that they scoop the environment there, and they sense danger. They prompt also this other, part of the army, this one in blue, they are the B cells, to be ready to deploy their weapons, i.e. immunoglobulins, that they sometimes can be deployed outside the wall of the city, so there will be the security IgA that will come over here and fight the enemy outside of the wall. And then there is this one in white, they are the T cells that eventually will move to the next level, so this cell-mediated immune response, integrating this innate versus adaptive immune response and so on and so forth. But the message that I want to tell you, pass along here, see how complex and sophisticated is the immune system just underneath this single layer of epithelial cells, and that speaks volumes of this, decision-making process that we do every day that we are totally unaware, the balance between, tolerance, i.e. state of health, or immune response, that it means inflammation and therefore disease. Now, we know, for example, that in even autism, there are GI disorders, and therefore these battlefields that eventually will lose tolerance, that will develop immune response, can create the collateral damage that we call inflammation. But how frequent are GI problems in autism? Well, there is a tremendous window of numbers that you're going to see there, mainly because we have no idea for some of these symptoms. When they are objective, so in other words, they're not be reported by the individual, but you can measure objectively, then we have a better idea. Constipation between 6 and 47%, diarrhea 3 and 77%, belching, vomiting, and so on and so forth. Still, these are objective, so you know that you see the poop there. You can't establish if there is diarrhea or not, and there is such a window of variation there. Imagine when we talk about reported or suspected signs and symptoms like stomachache, discomfort, enteric infections, gastritis, and so on and so forth. The kids, particularly the ones who are not verbal, they can't communicate. They can act weird or they are in distress, but if this distress is due to colics or reflux or nothing to do with the GI tract, it's totally unknown. And that's the reason why you see here all these question marks, because we have no idea. So what do we know about the pathogenesis? Now, interestingly enough, again, slowly but surely, and it's not accepted, as face values by all scientists, but people that start to really come up with this concept that autistic spectrum disorders are not different from many other multiple factorial disorders like, autoimmune diseases, cancer, and so on and so forth. They are absolutely undisputable genetic elements in autism. I don't think that this is object of discussion anymore. The fact that the environment played a tremendous role in the pathogenesis of autism is also out of discussion. What is coming really more and more, interesting is this loss of barrier function, the mucosa barrier that keep this uncontrolled, passage of enemies coming in, so that, on a specific genetic background, that can impinge on the immune system, so that, this individual will lose tolerance and create inflammation that eventually leads to the problems that has to do with digestion, with the brain, immunity, heavy metals, and you name it. And the list goes on and on and on. So what are the evidence of these three elements that are involved in there? Well, genetics now with GWAS and many descriptive studies and mapped out a series of genes that have mutation that seems to be cluster associated to autistic spectrum disorder. So these are the linkage disequilibrium studies done. This is some of many studies all put together. But it's interesting, of course, each of these red line it's a gene in which has been found a mutation. Some of these mutations are in structural genes. Some are, in introns, so the non-coding regions. But nevertheless, you see how this is distributed. There are some chromosomes, like chromosome 7 and chromosome 16, in which there's a major cluster of genes that have been found. Some others, interesting enough, chromosome Y seems to be spared of these mutations, and so on and so forth. So definitely there is a genetic component. We knew this from twin studies, for a long time ago, so the genetic component seems to be undisputable. Why environment? Why the environment seems to be a big deal? Well, again, there is a syndicate of evidence. They are very circumstantial, but pretty strong. The environment has something to do with the outcome and the pathogenesis of autism. Definitely, there are increased concern about environmental exposure in neurodevelopment in general. And again, who does this for a living? And I do not. But today and yesterday, I had the chance really to indoctrinate myself. It seems that, again, this is not premises just of autism, but any other neurodevelopmental disorder seems to have an environmental component there. There are, provided data on mechanisms by which environmental exposure may cause autism, and there are some interesting animal models that are applied here at the Mind Institute and many other places. Definitely there are epidemiologic data that seems to suggest that there is an environmental element there. There is a single genetic model of autism. In other words, that you mutate a gene and 100% of the offspring of that particular breed of animal, whatever, will translate in autism. And most importantly, there are this descriptive data, this epidemics. We're in the midst of an epidemic of autism that is quite remarkable. Among all the environmental triggers, the ones that I'm interested the most, not because I believe they are more important than others, but because it's in the realms of my expertise, are nutrients, and particularly gluten and casein. And that one of the most popular interventions that is purely based on anecdotal data, and there is a tremendous amount of discussion that toward the end we can revisit, is the benefit and the efficacy of a gluten-free, casein-free diet. But on a large scale, by far, this is the most embraced treatment in kids with autistic spectrum disorder. What is interesting about gluten, believe it or not, is the fact that we as a species, human beings, were not built to deal with this. And when I say we, I mean we all. As a matter of fact, for the 2.5 million years of evolution, 99.9% the humankind have been on a gluten-free diet. Gluten came only 10,000 years ago when our ancestors changed dramatically their lifestyle from nomadic, i.e. chasing food and seasons of stables, to settlers. And they start to play with, grains. And this tribe, the Ordi, to which wheat, rye, and barley, the grains that belong to which they contain gluten, it came about only at that time. So we grew up in terms of a species with all these other tribes of grass family, like rice, oats, finger millets, teff, and so on and so forth. They are all gluten-free, and this just came at the very end. So biologically, we're not built to deal with gluten. What that means, that, you know, as you will see in a moment, we're not able to make, you know, use of gluten and digest in the proper way with some consequences. So we look at the genes, we look at the environment. What about this intestinal barrier business? That was a black box until the recent past. I mean, you know, this issue of the leaky gut has been vilified at the wazoo, particularly by, you know, not really, how to say, well-controlled studies, mainly in the alternative medicine world. That, again, I have full respect. That's not a criticism to alternative complementary medicine by all stretch of imagination. But again, it looks like that everything that was wrong in life was due to leaky gut. You know, the fact that you are in a drought is due to leaky gut. The fact that we are -10 in Boston was leaky gut. You know, that you are in an economic crisis in California, leaky gut. But anyhow, so what are the facts? The facts are very few. There are a lot of fantasies. The facts that we deal with a very long tube. In an adult, it's roughly 20 feet long. Covered, as I show you at the beginning, by single layer cell. That's it. That protect us against what is out there. Physiologists, gut physiologists in the early 80s conceptualized this single layer cells like a floor in which each cell was a tile, and there was cement, grout, that sealed everything in between. So anything that pertained this interface between environment and the host through the GI tract had to be negotiated through the cell. So stuff coming through has to come through the cell because there was no space in between. And we're talking about a huge interface. You take a gut and an adult, you stretch on the floor, and you core a double tennis courts, 3,000 square feet. So imagine the amount of exchange of information on a daily basis. And again, nature did that to put all this, you know, stuff in the small area of the belly, not just because we want to digest well whatever comes there, but because we really want this interface. We really need to really have this continuous exchange with the environment to really shape the way that we are and our many functions, particularly the immune functions. So a dramatic shift of paradigm happened in the late 70s, early 80s, when a Japanese group said, you know what? The space in between cells, no grout, no cement, no wall, not at all. They are doors, almost closed all the time. But they are doors that imply that they can, under specific circumstances, being open by signals that we didn't know for a long time. So why we have these doors? Because we have a tightly controlled antigen trafficking in between cells, because we want that. That's something that we definitely want. That's how we, bottom line, we induce tolerance by doing antigen sampling that way. And over the years, the complexity of these, you know, structures that technically are called tight junction increase exponentially. And, you know, there are structural, you know, proteins that really make the door like occluding, that there's a Claudin family. There are 17 now that I will tell you later on. There are a cadre of proteins in the cell, the cytoskeleton is involved, and so on and so forth. But again, from the early 80s until 2000, we had no idea what was the key that was regulating the permeability of these gates until we stumbled upon, by serendipity, to this molecule called zonulin that remains so far the only physiological modulator that makes this tight junction to loosen up on a very short-lived signaling. So it takes a few minutes to open up, a few minutes to close down. If zonulin is released under physiological circumstances. So that's pretty much what we know about these tight junctions. As typically happens in physiology, you push to the limit, you spill into pathology. And it's now pretty much obvious, because there are many studies from many groups worldwide, that these gates that open and close in response of many internal and external stimuli are physiologically involved in immune surveillance, immune functions, regulates the exchange of small molecules, protein, and cells across these barriers. And now what we believe that was the premises of the intestine up in the kidney, up in the blood-brain barrier, because they all function the same way, structured made the same way, they are regulated the same way. And this brought also to a paradigm shift in terms of many inflammatory diseases in which these junctions seems to be dysfunctional. Rather than to have this tightly regulated trafficking, now they loosen up their control. So these gates, rather than to be closed all the time and only occasionally open under zonulin, signaling and then close immediately, they got stuck open. Many of these conditions because, as you will see in a moment, because of upregulation of zonulin. So zonulin, this molecule, was discovered for the first time in 2000. We realized what kind of molecule we're talking about after almost 10 years. Many postdocs' careers were burned on the altar of finding what the heck this molecule is all about, because it's a weird, strange protein. It turns to be the precursor of a molecule that is very ancient, it's called Haptoglobin. Haptoglobin comes in two shapes, on two flavors. Haptoglobin 1, that was the oldest of all, it came very, very early, roughly 450 million years ago. Soon after that, fish split for the rest of the animal kingdom. So fish, they don't have haptoglobin. And for many, many years in clinic, haptoglobin was used as a biomarker of inflammation. Before the descent rate and CRP were used, haptoglobin was the marker of inflammation. What the haptoglobin does for living? Scavenge hemoglobin. So when you have hemolysis, in other words, this massive destruction of red cells and hemoglobin comes out of the cells, if not taken out, will eventually induce oxidative stress and therefore death of tissues. So haptoglobin is in charge to take hemoglobin out and prevent that to happen. Many, many moons later, 2 million years ago, 500,000 years after humans split from chimpanzees, this second haplotype, the haptoglobin 2, came about. As a matter of fact, this was due to an unbalanced crossover of one of the subunits of this molecule, and this is slightly bigger than this molecule here. But what is important for the discussion here, that zonulin is the precursor of haptoglobin 2, a molecule that should not do anything in life, because as precursor, by definition, should be inactive. But turns to be a molecule that in its pro form is able to loosen up tight junction, and when cleaved does the business that I was telling you before. And again, there is a rationale in terms of innate immune response and prevention of oxidative stress that makes a lot of sense, that you have dual function with a single protein. Buy one, get the second free. So that's been one. But also it's very intriguing that in its form as pre-haptoglobin 2 precursor, alias zonulin, only humans can do that. By the way, how many autoimmune diseases afflict humankind? Any idea? 100? A little bit less than that. Roughly 65, 70. How many spontaneous autoimmune diseases are affecting chimpanzees and primates in general? 0. Yep. There is no autoimmune disease, spontaneous autoimmune disease in chimpanzees. So the location of the gene for zonulin is also interesting. It's sitting on chromosome 16. And again, the evolution of the zonulin is quite interesting, but it's sitting on chromosome 16 that is very dense of other genes that are associated with autism. And matter of fact, since this has been published, and this was in 2010, so not long ago, a large number of individuals have been studied zonulin as a biomarker for possible diseases. If you look at the chromosome 16, quite an interesting chromosome, tiny chromosome, 3% of the human genome is encoded over there, roughly 1,300 genes, but genes of 3 major conditions, category of diseases are sitting there. Autoimmune diseases, like, for example, inflammatory bowel disease, lupus, type 1, rheumatoid arthritis, cancer, many leukemia, breast cancer, Fanconi's anemia, lymphoma, prostate cancer, and disease of the nervous system. Lou Gehrig's disease, leukodystrophy, MS, autism genes are sitting here. When the gene for zonulin was identified and located in chromosome 16, again, many individuals and many centers study this as a biomarker, and sure enough, clustered with the same three categories: autoimmune disease, ankylosing spondylitis, celiac disease, Crohn's disease, rheumatoid arthritis, and so on and so forth. Cancer, particularly gliomas. There are studies from Germany, quite elegant studies, showing that the more advanced is the state of the glioma, the more the blood-brain barrier is breached, the more zonulin is expressed. Breast cancer, lung adenocarcinoma, and so on and so forth. And again, this is the nervous system, including chronic inflammatory demyelinating polyneuropathy, MS, and schizophrenia. Nobody studied yet, autism and zonulin expression. So, what are the environmental triggers that cause zonulin release and making the intestinal leak? And again, in terms of autism, in terms of leakiness of the gut in general, I want to really focus on two, i.e. grains and the microbiome. That's the ones that I really want to focus the discussion. I was telling you that, we're not built to eat, gluten-containing grains. Why? Gluten, like any other proteins, can be compared to a sort of pearl necklace. In order to make use of proteins, we have to break this necklace and then cut this in pieces that we call technically the peptides, and then we peel off one at a time of these pearls. They are the amino acids, so that we can absorb them and then make use of them, either burning them for energy or rebuild proteins based on our program. We can do that with pretty much everything that we put in our mouth, but this guy here. The best that we can do is to break in pieces, but we cannot complete the digestion. Why? Because the composition of this protein in terms of amino acid sequence is very weird. It is extremely rich in proline, there are P's, and glutamines, there are Q's, and there are so many in here that our digestive enzyme, both pancreatic enzyme and brush border enzymes, they can't do that. The best that we can do, again, is to break in pieces, but can't go any further than that. And when you have peptides, and eventually these peptides will make the journey on the other side of this barrier, they can be seen by the immune system as dangerous signal, as enemies, as non-self, and you can mount an immune response. And these peptides, they are undigested. They can do different things. For example, this one that was described, in the 90s, induced apoptosis, programmed death of cells. Not in celiac, anybody. This guy here, this yellow one, is a super antigen that induces an inappropriate, exaggerated immune response when exposed to the immune system of anybody. These two guys here, they bind to a specific receptor and induce the release of zonulin. And so in other words, these two peptides make the intestinal leak so gluten can get access underneath this barrier. This is the last guy that was identified in our lab that is capable to induce recruitment of neutrophils on the battlefield. So this is, in other words, is able to release IL-8 and consequently neutrophils. They come in the lamina propria of the intestine, ready to create inflammation. So bottom line, gluten can cause leakiness in everybody. You eat gluten, gluten peptides bind to this receptor, release zonulin. Zonulin binds to this machinery, then instruct this space in between cells to be open. It comes over here. What happened after that depends who you are. Vast majority of people will clean the mess, will never see this, will be totally unaware, and we stay healthy. Some people may eventually develop food allergies because now you have a leaky gut, there are undigested food stuff here, and you develop allergies. Other people, they can develop autoimmune diseases. And all depends on what kind of genetic background you have. Now, that this leaky gut is part of autism has been, a discussion that is far to be settled. This is a paper that was published a couple of years ago from a Napolitan group in which they look at the permeability of the gut with a rather complex assay, this lactulose mannitol test. So compared to kids controls, autistic kids, they have a leaky gut. Interesting enough, their relatives have a leaky gut compared to adult controls, meaning that a breach of the barrier may be necessary, but not sufficient to create the problem. Because again, depends what kind of immune system you have on the other side of the barrier. When these kids, they are on an unrestricted diet, they leak. When they go on a gluten-free diet, their leakiness goes back to normal compared to normal kids. So it looks like that, again, is the exposure to gluten that seems to make this intestine leak, most likely because of this issue of these peptides that make the intestine to release zonulin. This is a very busy slide, but the juice of this is that if you look at many antibodies, because remember, when you breach this barrier and stuff that doesn't belong to our body comes in, the immune system sees these enemies and starts to deploy weapons, i.e. antibodies, against these specific proteins. In this case, there are antibodies against gliadin, that is the component of a gluten that is more immunogenic. And if you look at the IgA and the IgG, particularly the IgG, you see that the kids with autism, they have an increased level of IgG anti-gliadin antibodies that goes back to normal, when they go on a gluten-free diet. And again, this created tremendous confusion because the people, they say, the positive anti-gliadin antibodies, that means they have celiac disease, and therefore, that's the reason why they have to go on a gluten-free diet. Absolutely incorrect. The presence of these antibodies mean only three things. One, this individual was eating gluten. Two, there was a breach of the barrier. Three, gluten came in, and the immune system did its job, mount an immune response against a non-self antigen, and that's all. And when you take this out of the diet, it goes back where it's supposed to be. So that's what it means. But this is another indirect way. Matter of fact, some gut physiologists, they use anti-gliadin and IgG antibodies as a proxy for gut permeability. And again, the same happened with the total IgG, the milk IgG, you see they go down, Casein, the same story. So, and again, it's obvious. You take this out, and the immune system has no enemy to fight. These antibodies, they go down. So what about the gut microbiome as the second instigator of, impairment of the gut barrier and all 9 yards? That's an interesting story because, once again, remember, guys, I'm talking about science that really materialized in the past few years. As we didn't know too much about the gut barrier and its regulation until 20 years ago, this microbiome was a black box for many, many years. And now we have the tools to study and therefore makes some sense about this. So it's now clear that we have a parallel civilization that live with us from birth to death. They've been overlooked for many years. That seems not just sitting there doing nothing, but has a great deal of what we are and how we function as human beings. And matter of fact, the complexity of the human microbiome in the mouth, and then the skin, and then the genitourinary tract, but most importantly of the intestine, which there is the most rich microbiome, now is taking shape because now we have the tool to study it. And then we have the understanding what is the composition and how this composition changes related to specific condition, for example, autism. So what I learned about this is the following. Mainly because we decide history on the face of the earth, we decided that we are definitely the most complex species on the face of the earth because we write the textbooks of history, okay? I don't know about you. But when the human genome was completed, at that time, we realized that we're genetically rudimental. We're made by only 30,000 genes. Now we are downgraded to 23,000, 24,000. Let me put this in perspective. The worm, the one that we dissect in elementary school, 90,000 genes. Wow. The genome of the plant that makes gluten, wheat, 150,000 genes. 99.9% identical, not similar, identical to chimpanzees. I also work with chimpanzees like you guys. I can't talk about politics with them. Got upset because they were late for lunch or dinner. I don't have an intelligible discussion with them. And yet only 400 genes make the difference between us and chimpanzees. How is that possible? How this is really conceptualized as a possibility, unless you consider the fact that we live in symbiosis with a parallel civilization that complexly generate 100 times more genes than we do. And I have a hard time to believe that these neighbors, this civilization that has been here millions, billions of years before that we materialize as a species, and when we'll be so nuts to extinguish ourselves that we continue to live, would not talk with us, just sitting there and not have any communication with us. That would be hard to believe. So this is my interpretation of the story. I believe that we are really made by two genomes. The human genome that is inherited by our parents, that is stable, will never change in its composition. Let's say that intrinsically has some defects. Genes that make us a risk to develop autism, okay? The fact that you have that genes there is not implicit the concept that it's destiny that you will develop it. You have the potential to. But if you develop or not, autism depends on this other genome, the microbiome, that if we're lucky, comes from our mother. I.e., if we're born by vaginal delivery, comes from our mother, and because this microbiome live in peace with her, will most likely live in peace with us. It's extremely dynamic, change over time, change from one individual to another, and the same individual over time. So this can explain the inexplicable. For many conditions, like many autoimmune diseases for which we really didn't have an understanding how all this can happen. So we know, for example, in celiac disease, that if you're born by C-section, your chance that you develop celiac disease is 3-4-fold higher than you're born by vaginal delivery. Now, we had a chance, as Judy was telling you, I was just pulled into this, with Paul Ashwood having the lead, is right there, of being supported by the Autism Spectrum Trailblazer. And Paul was wise and visionary enough to say, you know what, this takes a village. So he put the syndicate of people around in which he, expert in immunology, the folks at Caltech, expert animal model of autism, and again, the B. fragilis stories and so on and so forth, and us with gut biology and immunology to try to really answer these questions, i.e. to define the biology of the gastrointestinal dysfunction in autism spectrum disorder, using both the animal models and clinical trial. And the paramount goal that I think that you will see will have a lot of sense now is really to provide a rationale for novel personalized treatment for autism. Because one thing that I realized coming late and totally being not good with autism, because I don't understand a squat about this, is there is this expectation from many constituents that despite this is multifactorial, despite there may be multiple causes, everybody's looking for this single magic bullet that will fix them all. And I hate to say that, and the bad news, that is not going to happen. So Sarkis, and again with the support of Paul's grant, just published in Cells this amazing, interesting results on this mouse model, the poly-IC that many of you guys are familiar with. And they look at different parameters, for example, behavior. And again, the center duration, how bold are these animals rather to stay on the edge of the cage, to stay in the middle, that should be totally unrealistic. This, repetitive behavior, marble bird, and the total number of calls. Bottom line is that these animals, these are the saline, this is saline plus the bifidobacterium. But if you compare this column to this column, these are the poly-IC, you see how they are worse in terms of center duration, worse in terms of marble bird, that they do much more, worse in here, and all is fixed by giving these animals B. fragilis. So in other words, that these animals end up to have dysbiosis, so change in the composition of the microbiome with the underrepresentation of the species B. fragilis, and resupplying B. fragilis fix the problem. Now, even interesting is the gut permeability that again, with this chemical that is called DSS, we know that you can make this intestinal leak. This is the saline, and the poly-IC, they have the intestinal leaks that is fixed by B. fragilis. And this was parallel with the change of expression of a series of this component that you will see in a moment, this tight junction protein, they seem to change, in terms of, when you got autism with the poly-IC and when you're fixed when you got, B. fragilis. Extremely elegant. The question again, can we extrapolate this to humans? Because after all, these are mice. So, there is a leap of faith here. Now, what about in children? And again, here is something that Paul and I have been working for a while, and the goal is to identify, I forget about this jargon. The bottom line, when we talk about microbiome, and again, I chat with the students before, it's a generic, concept, but, compare the microbiome to a sort of a let's say, a town with different neighbors. So there are the Italians, the Jewish, the Greeks, the Irish, and so on and so forth. And they live in peace, provided there is an equilibrium. That is what is the microbiome. There are these different species that live all together. And that's pretty much what the microbiome is made of. Now we can do stuff that were unthinkable in the past. We can answer 3 different questions. They are absolutely instrumental to understand how the microbiome can impinge on the human genome so that you change your immune behavior and you go from tolerance to a disease status like, autism. Number 1, who is there? So you can really take a look and say, who's living in this village? Number 2, what is the distribution of these different neighbors? So how many Italians versus Greeks versus Jewish and so on and so forth? And 3, and most importantly, that's something that was unthinkable till 2 years ago because technically unfeasible and extremely expensive to ask what they're doing. In other words, what kind of genes they are expressing so that we can explain how this can impinge on the behavior and the function of our genes if they have an impact on that. Now, the other confusion is when we talk about microbiome, what we're talking about. There is a hierarchy here. So that it goes from the domain that is the universe to the kingdom. So we're talking about bacteria, and then there are phylum, class, or the family, genus, species. So when you talk about microbiome, the vast majority of analysis is at the kingdom level. So you see that, or even better, at the phylum level. So that's pretty much what you can do. It takes a little bit more sophistication to reach all the way to the specific stuff. I give you here an example. When you talk bacteria, there are all the bacteria. Then the phylum, you can talk about the specific phylum, like the Firmicutes. And within the Firmicutes, there are the Clostridia. And within the Clostridia, there are the Clostridiales. And within the Clostridiales, there are the Raminococci. And then within the Raminococci, there are Anerotrungus. And within the Anerotrungus, there are Anerotrungus species. So that's the complexity. Did you got anything? No. That doesn't matter because I don't understand myself. So that's okay. But all this to say that, again, you got to be very careful when you talk about microbiome because, this is like to say humans, Americans, Californians, North Californians, nearby the border. Eventually, you talk about, Sacramento and the neighbor that is nearby UC Davis. That's what it is. So when you say humans, you can, judge mode there. The other thing that is important that it's become extremely clear that now these 2 phyli, the Bacteroides and Firmicutes, that makes the bulk, almost 90% of our microbiome, are extremely important. In general, with a lot of exception, these are the bad guys. So when they increase, this is bad news. Meaning you lose tolerance. You start to have inappropriate inflammatory response. These are the good guys. These are the ones that keep us in check. When we look at the microbiome at the phylum level, we look both at the ratio between Bacteroides and Firmicutes. And the other thing that in general we look at is the diversity. The more diverse is the village, the more neighbors there are, the more healthy is the microbiome. The more homogeneous, the less diverse, the more monotone it is, the more likely that you are off balance, i.e. you have a dysbiosis that can create problems. Now, this is the heat map of some of the samples that we have been looking at, and Paul sent us mainly 4 categories of kids. Kids with autism and GI symptoms, they are the ones in red, these guys here, 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 blah, blah, blah. The autism with no GI symptoms, they are the one in green, And then healthy control, they are this pink here. And then this in blue that we have very few, they are the ones that are healthy control with GI symptoms. And the heat map is telling you the abundance of all this crap of stuff here that are mainly at the species level of the different species. And it suffices to say that, you know, the darker the color, the more abundant it is. And again, the bottom line, the analysis I've done so far, it looks like the only thing that sticks out is that the autistic kids with GI symptoms, they have more bad guys, i.e. bacteria in the family of Firmicutes than all the others. And they, in general, this alpha diversity, it's just to compare the microbiome, i.e. the village, of one group versus the other. You see there is a pretty good overlap between healthy kids with or without symptoms and autistic kids with no GI symptoms compared to the ones that have GI symptoms. They distinguish them some from the others. So there is a diversity of the composition, this village of the kids with autism and GI symptoms. And this is another way to see this, how diverse is the village. And these are the autistic kids with GI symptoms versus the one with no GI symptoms. And these are the two controls. You see that they look as median very similar to this that seems to be diverse. This is another way to see the diversity. And again, this is very colorful, so that's the reason why I like it. You see here that the red dots, they are the autistic kids with the GI symptoms. They tend to cluster in the center. So in other words, they are much similar to each other. While the autistic kids with no GI symptoms, healthy controlled, they are in green, they are more dispersed. They are everywhere. So there is more diversity here. There is less diversity here. And I told you, less diversity is bad news. Because, you know, the less diverse you are, and the more likely that you are off balance and you create problems. So when you look at the Clostridia bacteroida, Clostridia belongs to the bad guys, i.e. the Firmicutes. Bacteroides belong to the good guys, to the Bacteroides. You see that in an autistic kids, the balance is significantly skewed in that sense. So this is the working hypothesis, and so this is our working hypothesis. So we have a situation in which these kids are born genetically skewed to develop, autism, okay? Now they have a situation in which, because of nutritional issues, because of, environmental pressure that, again, nothing can be excluded, in my humble opinion, can be exposed to metals, additives, colorants, you name it, whatever you like it, that can, antibiotic treatment, infection in the mother during pregnancy, anything that can really create a situation of dysbiosis, and that create a breach of this barrier. Crap stuff start coming here. So this non-self-antigen, including gluten, casein, and God knows what else, bacteria byproducts, got access to the lamina propria. There, you activate the inflammatory cells that cause local inflammation that, if they stay there, so in other words, when the cells, they see the enemy, they're activated, they develop and deploy weapons, there is a collateral damage that we call inflammation. If they spend most of the time there, the vast majority of the cells stay in the intestine, they create a local inflammation, translate in GI symptoms. Now, I told you that the intestine is not like Las Vegas. Sometimes cells primed in the intestine, they leave the intestine and are programmed to go somewhere else. And eventually, they migrate in other districts like the brain, where they can cause local neuroinflammation that, on that specific background, can translate in autism. So this is the overall hypothesis. So again, I've been discussing the Paul and many other folks this so many times. Let's make numbers so we understand each other. Let's say that this cascade activated 1,000 immune cells. And let's say that these immune cells, they create the steady inflammation because they start to produce these chemicals, they're called chemokines and cytokines, that induce this collateral damage, this inflammation. If all 1,000 cells they are in the intestine, they stay in the intestine, you have GI symptoms and nothing else. But let's say that 500 of these cells, once activated, they are programmed to leave the intestine and to migrate in the brain, and now they're creating neuroinflammation. These are the kids, autistic kids, they have both GI symptoms and behavioral symptoms that put them in the autistic category. At the other extreme, you have the activation of these cells all 1,000 cells, once activated, will leave the intestine, all will go to the brain, and those are the kids with autism without the GI symptoms. That's pretty much the working hypothesis. What kind of elements do we have really to support this hypothesis? Very little. But we're working on a syndicate that come, to fruition with some of the stuff that we're seeing. So how we put all this together, how we can really, make the story that will make sense. And again, this comes mainly by studying autoimmune diseases for which we thought that we have the entire story straight. We can compare the human genome to a sort of piano with 30,000 or 23,000 notes, each of them, encoding a gene. And let's say that theoretically, 300 these notes, if stroke, they will play the song ASD. So if nobody's sitting this piano, there is no music that comes out of there. The piano players are indeed is the microbiome, that it can be extremely dynamic. And depending who is sitting the piano, you have different music. Let's make the assumption that Elton John is the one that is playing the piano. And therefore, he is able to play tunes in which 2 of the 300 notes that belong to the autism song are played. There is no autism song played. Now, let's make the assumption now that something happened. Antibiotic infection, vaccination, you name it. They changed the microbiome. Now it's Ray Charles sitting down. Ray Charles doesn't play pop music, rather jazz. Jazz will require all 300 notes to be stroke. And now you have the song Autism that will be played. That's the only way that I can explain why there is no 100% concordance in identical twins. And that's also can explain why some kids develop autism at 6 and others they develop at 16 months of age. And for example, in celiac disease, there's the extreme paradigm. We know the trigger. We know that it's gluten. Everybody's supposed to gluten at baby age. When baby food is introduced, there are people that can eat gluten for 50, 60 years, not having problems, and then all of a sudden they lose tolerance. Why? Because that's what happened. Is this fantasy? Now we start really to understand this by studying the metabolome that is like reading music. So if I look at the metabolites, there are the byproducts of this interplay between human genome and the microbiome that activate the specific metabolic pathways, that create the specific metabolites. And I read metabolites that belong to this guy here. So I read the pop music. I know that I'm safe. I know that I don't have a problem. But let's say that now I see that jazz music is being played. I know that this other guy is sitting here, and down the road, the music autism will be played. That is the holy grail of preventive medicine, to try to customize treatment and intervene by reshaping the microbiome by using prebiotics or probiotics and put these kids back in a state of tolerance. We have this proof of concept celiac disease. We identified 2 metabolites that they change 6-8 months before that these kids lose tolerance to gluten and develop celiac disease. And now we're trying to expand this on a large scale to really make this a solidified, validated biomarkers. There are already some studies in using metabolites, controls, siblings, and autistic kids, and you see that they can really segregate so that you can figure out who is who just based on the metabolite profile. So I'm going to conclude with something that really struck me. Okay, when this gluten-free, casein-free diet became such a heated debate, to the point in which there was these 2 camps, the believers and unbelievers. The believers said that's the panacea. All cases of autism will be fixed with this diet. And the non-believers said, what a junk. This is absolutely crap. Nothing is going to work with this. The NIH came into the picture and said, enough. We want well-controlled, double-blind studies so you can give us an intelligible answer. Studies were done, a Cochrane analysis was run, and they identified 3 significant treatment efforts in favor of the diet intervention. 3 outcomes showed no significant difference between treatment and control. Split 50-50. And what was the conclusion? Research has shown high rate of use of complementary alternative therapies for children with autism, including gluten or casein exclusion diet. Current evidence for efficacy of this diet is poor. Large scale, good quality randomized controlled trial are needed. I like to say, this is garbage. You put more garbage, we will understand what's going on. The reality of the story, again, being totally unbiased, is that, again, we miss the point of the complexity of the matter. So I will give you a theoretical example that a biostatistician will appreciate. Let's say, theoretically, that the population is divided by different pathogenesis pathway that leads to the final destination, autism. You know, you can have intestinal dysbiosis, genetic mutation, oxidative stress, metabolic disorder, gluten sensitivity, immune-mediated, Fragilis, you name it, okay? Let's say that there is this, distribution. And let's say that I am part of that Cochrane analysis. I have 100 kids, and take this population, and put all of them on a gluten-free diet. And let's make the assumption that these 20% are the ones that will respond because they have the gluten sensitivity. Efficacy, 20%. Failure, 80% of the kids will not respond. So my conclusion is the gluten-free, casein-free diet doesn't work. Take the same population, exactly the same 100 kids, same distribution of pathogenesis, and now I have biomarkers so I can see who has gluten sensitivity, and I do the gluten-free diet only on this 20. 100% efficacy. Same population. All this to say, guys, and I was telling the students today, it has been an interesting journey of our, profession of medicine, no matter what kind of healthcare professional you are. 3,000 years ago, doctors were healers. They were treating the patients. They were looking at the patients. They were trying to, fix whatever method they have, prayers, look at the moon, giving herbs, whatever. Then we realized that this was crap, and we shift our attention from, the patients to the disease. We became very wise. We studied the mechanism of the disease. We want to be evidence-based because we made the assumption that autism is homogeneous, celiac disease is homogeneous, MS is homogeneous, and cancer is homogeneous, so that disease can be treated the same way no matter who you are. Now we realize that's not the case. And the pendulum is swinging back to personalized medicine. Healers, again, thank God, because that's what it is. This is, diagnostic, treatment, customized, so personalized medicine. So what are the takeaway message that I want to leave you with? Epidemiology. We have no freaking clue how often GI conditions are afflicting kids with autism for the reason that I tell you. So biology, there are several studies that seem to suggest that the immune system that is out of balance in autism. Some GI condition may have an immunological basis. Some individuals with autism may have abnormalities in serum concentration, neurotransmitter, serotonin, but God knows what that means. Serotonin can be associated with anxiety, so there is the possibility that the brain-gut axis is really driven by serotonin. That seems to be quite a feasible and plausible possibility. Nutrition, we discussed this at the wazoo. The treatment, there is no magic pill here that can fix the problem. What are the gap and opportunities? What is the pathology of gastrointestinal condition that you can really understand through integrated studies? The gut microbiome, the metabolism, the inflammation, the immunity, and mucosal integrity. That means it takes a village. It does not take the single individual. We have to understand, and that's something else that I told you guys during these 2 days. Being an outsider, I'm mesmerized by, one, the fact that we're in the midst of an epidemic. Two, that there is a surge of collegial efforts to really stop what is the travesty. And this would take not a single man. We take something like the Mind Institute, in which different expertise, they come under the same umbrella, coordinated in an effort to stop these epidemics. The study to manipulate the microbiome in animal models may help, but the animal models, with all the respect, do not reflect completely what is the microbiome of human beings. So definitely that will help. particularly studies done in primates, like Judy and her colleagues are doing here, but ultimately you need to do this study in human beings because you cannot extrapolate completely. Then the key elements is to identify and validate biomarkers, because that's the way that you stratify the population, and that's how you can do customized treatment. So I know there is a tremendous pressure from parents, and I'm very sympathetic. But again, science, it's an element of patience. You go step by step. You walk before that you run, because otherwise, if you run, you miss the turn and you go in dead hands. And that's frustrates everybody. I'm going to finish with a quote from Anthony Good. How many of you guys have seen this movie, Ratatouille? All right, good. For the few that didn't see the movie, this is a guy that was extremely skeptical that rats can be good chefs. God knows why he was skeptical. And at the end of the movie, he had to really realize that a good chef can come from unforeseen directions. And this is the quote that I really enjoyed. The world is often unkind with new talents, new creation, or new ideas. The new needs friends. And again, when you talk about shift of paradigm, the scientific community very rarely will embrace you as a visionary, will embrace you as a nuts. Not everyone can become a great artist, but a great artist can come from anywhere. So if we really are interested to really tackle and try to help resolving this problem, we need to take off our preconcept, just get undressed and put on the coat of humility, sit around the table and work as a team. Otherwise, this would not go nowhere. I believe that in 49 minutes, I was able to encompass everything that I really had to say. There is a lot of stuff that's going on, but I hope that I gave you a sense of two things, two messages I want to give to you. The brain and the gut, they talk each other. And the hierarchy of importance, the gut is much more important than the brain. Thank you so much.
I'm an adult with autism, so I thought you might be interested. I have had trouble as a kid with GI problems when I was a kid. And then October the 13th, I suddenly passed out. And I had to be taken to the Mercy General Hospital with an ambulance. And I have nothing wrong with me. So the doctor said I had a GI problem. That could be just a coincidence, or just, you never know. It could be eating spinach or something. But this has never happened to me before. And also another thing is I will say that one thing is I lift weights a lot. That has helped me with my stomach problems. And people believe that lifting weights does help with people with their stomach problems. Nobody seems to quite know why.
Yeah. So the challenge here to make a cause-effect link between GI upset and autism is that these GI problems are so common. And well, let me do this so that I can clarify the story. How many of you guys ever had a stroke? How many of you guys have ever heart attack? How many of you guys ever had pneumonia? How many of you guys ever had an episode of diarrhea? Did I make my point? Good. All right, next.
You talked about the ability to check for the genes for the gluten ability to, for the gluten and autism, the biomarkers. Is that something that the Mind Institute or different doctors are able to do, or is that something that's only at the research level at this point?
Well, the problem is when we talk about biomarkers of problem with gluten, there's been a lot of confusion. In the past, we thought that biomarkers means you have to be diagnosed with celiac disease. It's only recently that we learned that gluten can give you problem outside celiac disease, an entity that now is called gluten sensitivity. Right now, there are, possible biomarkers. For example, the anti-gliadin antibodies can be a biomarker, but never been validated. A biomarker that we validated now with autism or the schizophrenia is an antibodies against an enzyme that's called tissue transglutaminase 6. TTG, or tissue transglutaminase, comes in 3 flavors. TTG2, that is specific for the intestine, and that's the way that we do diagnosis of celiac disease. TTG3 that is specific for the skin, and TTG6 that's specific for the brain. Now, this is an enzyme that's within the cell. So normally, our immune system doesn't see it. You build antibodies against this enzyme only when you have, the cells that breaks out, it has a lesion, would die, so the enzyme leaks out. So when you have antibodies against TTG6, that means that you have neuroinflammation that is being caused by gluten. 20% of schizophrenic individuals that are TTG6 positive. Those are the ones that were targeted with a gluten-free diet with some primary remarkable results, i.e. that they resolve schizophrenia not with medication but purely on a gluten-free diet, but those 20%. We don't have that for autism yet.
I have a son with autism, and we took him off of gluten, and he's been on a gluten-free diet now for probably about 8 years. And we also took him initially off of casein, but we've let him eat some dairy products since then. Do you think we're doing the wrong thing? And I also don't understand what the coincidence of the sensitivities to these two things is in autistic people. Maybe I should have gotten that from your talk, but I didn't quite glean it.
So let me answer the first question. There are 50 plus treatments that are conceptualized for treatment of autism. Definitely the gluten-free, casein-free diet is the one that has least problem in terms of, creative problems, theoretical damage and so on and so forth, because after all it's the diet. And if you see the return on investment, by all means, I believe that you did the right thing. That's the bottom line. It's not that you deprived your kids God knows what. Matter of fact, nutritionally speaking, gluten is useless, mainly because we can't make use of it. The second question was, again, the second part of the question. That's right, how this works. Right. That's right. Well, there are two theories not mutually exclusive. There are some people that believe that some of these fragments, they are undigested, both of gluten and casein. They have a structural similarity with chemicals that control our behavior that we call endorphins. So glyendorphins for gluten, casomorphin for casein. And some people believe that these undigested fragments, they move and they go to the brain and they change the behavior of the kids on a specific genetic background because they mimic this function. Other people, and I personally tend to lead on this second theory, believe more the neuroinflammatory theory. So in other words, that these cells, they are in the gut, they're activated. Once activated, they move, they are programmed to migrate in the brain, and they create damage there so that you have this neuroinflammation translate and change in behavior. The reason why I believe this, because, some studies on brains of kids with autism, there are, brain bank and so on and so forth, they found cells, immune cells in the brain that were primed, i.e. came from the intestine. So that's the reason why I believe that that's a distinct possibility. Ma'am.
I'm a vegan, and I've learned that actually dairy has got to be the worst food. I don't know the man who invented drinking dairy. What was he thinking? He must have been fulfilling some sick mental, I want to go suck on an udder. No other creature on earth does that. Okay, and I was just saying I wanted to make, first of all, and I love Rejuvelac. There's this, that's the other part of the thing I wanted to mention. I said, how can you take away Rejuvelac? And that's actually good. There's wheat products like that actually won't give you the same, that would be good for your gut and won't give you the same reaction as bread will.
Yeah, and again, I would not criticize or dispute or agree because honestly, my honest answer is, I'm not sure, I don't know. Definitely, we human beings are not built to have the same milk that, a cow has. But again, we know that we can pay consequences if we're on a specific genetic background. But there are gazillions of kids that they thrive on a formula for a first year life and nothing else without having problems. So, but no question, and again, I want to make this clear, that if there is a choice, breast milk is the way to go. Now, if you want to become vegan as an adult, and therefore eliminate the series of proteins that you believe that don't belong to your body, by all means, I don't see anything wrong with that. But again, to go from there to say that casein is detrimental to everybody, with a huge number of people that they eat, casein-based products, so I mean milk and dairy products, it's really a leap of faith. So that's the way that I see it. But, yeah.
Curious your opinion about the increasing use of antibiotics and how antibiotics might lead to this dyssymbiosis, and also in the immune aspect, you talked about the antibodies, maybe if the children with autism have decreased amounts of IgA, which is supposed to be non-inflammatory.
That's right. So again, when lawyers, they talk about, particularly lawyers that deal with the penalty word, they talk about when there is an homicide, circumstantial evidence. You don't have the weapons in your hands, but there are so many circumstantial evidence that come over there. When you talk about antibiotics and their role, not only in autism, but a wealth of immune-mediated diseases, the evidence are quite compelling. These epidemics of immune-mediated diseases, autoimmune diseases, including autism, really start with the promiscuous use of antibiotics. There are evidence in the animal model that can happen. Definitely, we know that you take one course of antibiotics, and you change completely the microbiome, and this to rebalance, to go back to where it starts, takes months, not weeks, not days, months to go back. These epidemics happen only in the quote-unquote industrialized countries where antibiotics are used much more. We eat, sometimes, meat that have been, biffed up by antibiotics so that they can grow fast, and that's what we ingest. And I can go on and on and on. So definitely, there is a role. But by saying that this is the only reason why we got dysbiosis, I think that that's really, again, another stretch. I believe that we mentioned before in our private conversation, your grandparents, to go from Sacramento to Boston would take 2 weeks and would take time. Now you go back and forth in 24 hours. Imagine what happened to your microbiome if you, let's say, go to the other side of the world and come back. We don't use produce that are made locally anymore. And another example that I gave, is, when I was a kid, I ate strawberries in July and August. Now they are around 12 months. They are the same size, the same color, the same shape. Sometimes they come from Mexico. There are bacteria on there that are non-belong to the ecosystem that we have here. All this, can have an impact.
I have a question about the research or potential research that's done on zonulin, and whether there is any research in perhaps a pharmacological treatment to downregulate the expression of zonulin.
That's right. So zonulin was discovered in 2000, the gene in 2010. A spin-off company, University of Maryland, to really do exactly what you said, was created in 2005. The company now used as a prototype disease, celiac disease, because there are so many things that we know about this, and you can turn on and off the disease at will by giving the patients gluten or not. This zonulin blocker that will stop the overproduction of zonulin has been given roughly to 900 people. And 5 years later, $185 million later, strong safety data. This is a pussycat. This is a molecule that works in the intestine, so it's not absorbed systemically. And a very good, efficacy data. So people with celiac disease that eat gluten, they have symptoms. Gluten plus this blocker, they do not. And they don't mount an immune response. There is a final phase 2B that is concluded just now. Now they have to analyze data with 300 additional patients. That would be very interesting to know how this spin out.
Hi, I'm speaking as a mom for 2 milk intolerant kids. And I basically, there wasn't any research, there wasn't anybody telling me really how to take care of them. What I found was that when I baked the dairy, they could not have GI upset. Raw dairy would be 100% upset every time. I have one child who's now a teenager who loves to cheat, and it shows all over her face.
That's right.
And it shows with the time that she spends in the bathroom, and she's like, I'll deal with it, Mom, I'll deal with it.
That's right.
I get a little concerned, her abusing her body. Is this eventually going to lead to this autoimmune response later in life? And this, eventual allergy. I mean, they've been formally tested. There's a negative allergy, but I have lingering symptoms because my mom is now she's just been tested for her immunoglobulin. She has a very similar history as my kids.
That's right.
And she's now tested and her immune system is at 0. And she catches everything that is in every public space, and now she's more isolated, just trying to stay healthy for the
That's
winter. Right. And I'm wondering if these two are connected, these
That's
two
right.
generations.
So a reaction to foodstuff, let's say, milk and dairy products, can be on several bases. Allergic is one of them. You can have also allergy without production of those antibodies that make you diagnosed with allergies, like IgA antibodies. So you still can have allergy. But there are other forms of reaction or sensitivity to foodstuff. I mean, this is undisputed because you have a cause-effect relationship. She eats the stuff, and she breaks out and have GI upset. So there's no question about that. To have that kind of outcome, the immune system is acting weirdly. So in other words, it's seeing the enemy, i.e. the entire undigested products coming through and mounting an immune response. So she definitely has breached the barrier. She definitely has a genetic background that makes her, a risk to have this kind of reaction. If this is going to have long-term consequences, so it's just you pay the price on the spot, that there is no long-term consequences, it's hard to say. Because again, if it's purely an immune reaction to, milk and dairy products, the answer is just pay the price on the spot. There is no long-term consequences. But if she has also some other genetic background that will eventually, with this chronic reactivation and inflammation, can spill out of the intestine and the skin and go somewhere else, the joint, the nerves, it can definitely, come up with other consequences, but very hard to say.
Yes, I was just wondering about the inflamed immune cells and if in pregnant women they can trigger something in the fetal brain, and if there's been any work on that question.
Now, I'm not the best expert in the audience, but for what I read is, at least on animal model, there are very clear evidence that if you have activated immune cells in the mother, this can have an effect on the baby. And this can be either, indirectly or directly due to, let's say, an infection because the immune cells, they've been activated. One of the animal model that I show you was a model in which you give a byproduct of a microorganism. This will upset the immune system, that will create chemicals, and the offspring, they came up with this autistic-like behavior. So based on that, it seems like that indeed, if you have, a situation like that, you can really have, the immune system that can impinge on biology of the brain of the baby. There is also epidemiological data that, clearly demonstrate that, if there is a higher rate of infection in the first 2 trimesters of mothers that eventually will end up to have babies with autism. And this non-human primate model that they have here, they are studying, seems to suggest a very similar mechanism. And this is all mediated by these immune cells that are activated.
I was wondering what you think of the GAPS diet, because it seems like it's based on similar research, but it goes a little bit beyond just gluten and casein free.
That's right. So again, I have to tell you that as I am not really fond of the gluten-free, casein-free diet, just give it a try. I'm not fond of any kind of intervention like the GAPS diet, because again, the question here is, and the frustration is that sometimes, let's say that the GAPS diet, like the gluten-free diet, can be beneficial. It will be hard to, make the point that will be beneficial for everybody. The question is, who will benefit from the GAPS diet? If we continue to give it a try and put kids on different diets, on probiotics, put, on the bariatric chamber to do the detox, and you go on and on and on, the frustration will only increase. So to be honest with you, I would say theoretically may work, but for whom? And that's where the heart and soul of the question here. Again, trust me, I am totally sympathetic. If I was a father with the kids with autism and they told me that, to fix the problem, I have to jump out of the window, I will do it. I may ask which window, the one on the left or the one on the right, but I will jump. So I'm totally sympathetic. But this is now the time to sit back, reflect, and enjoy the fact that now we are in an unprecedented scientific position to start really to answer some specific question and not just blindly give it a try, but really have a customized, logical approach to the problem. I know that we want the solution tomorrow morning, and again, I'm totally sympathetic, but this, give it a try, it's something that I don't think that will be the logical way to go. All this long answer, not to say I don't trust the diet, I don't believe that it's right. It may be, but we don't know who will benefit from that.
So I wanted to ask, so for those, you said that some people can eat it 50, 60 years and nothing's happening to them. It did seem like avoiding gluten might be beneficial for all, since we never really had it in our diet 10,000 years ago. Now, for instance, for those who are eating it and it's normal for them, if they were to avoid it, do you think that their gluten sensitivity might increase with the avoidance of it?
Again, if you and I were sitting here, it's because thanks to gluten-containing grains. Otherwise, we're still jumping on the trees because, civilization came with agriculture. And now it is so entrenched in our life that, to say everybody goes gluten-free because otherwise we will be a extinct species, it's really a stretch. Numbers. How many people are on the face of the earth? 2 billion people, roughly, okay? All 2 billion, no matter who you are, even if you're Chinese, they're exposed to gluten. The number of people in trouble because they eat gluten, a fraction, a minuscule fraction. So, how we can impose a moratorium of gluten on the face of the earth when the return on investment would be limited to a minor fraction. It would be much more logical to say, for example, a celiac disease, to give you an example, you are Joe Schmo, 1% of chance to have celiac disease. If you are a relative of an individual with celiac disease, 10%. 9 out of 10, you still will be okay. If you have specific genetic markers that we know now, you go 20%. If you have 2 copies of these genetic markers, you go 35%. Now we can talk. Because 35%, I can sit and say, wait a second. Honestly, even if this is a pain in the neck, change the lifestyle, but I do a preventive intervention, I take that chance. Because 35% now is weight versus 1 to go on a gluten-free diet. But put everybody gluten-free or casein-free because, this is not going to, come up with something. Again, I have a hard time to justify that.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.